Okay, let's get started. Today we're gonna talk about epigenetics. So I started interested in epigenetics from it. Well, so actually, so this whole section, the second module, is kind of dear to my heart for the research that we are doing. And so remember, for gene expression, after you perturb the cell with all sorts of different conditions or drugs or developmental stage, you want to see what genes are regulated together. And the first thing we were interested in is to look at transcription factor binding. And we have work on this as well. And to find the transcription factor motifs. And then we realized actually for mammalian genomes or genome with a lot of enhancers that regulate genes far away, just to do motif finding would be insufficient. And so then we move to ChIP and ChIP-seq, and you can look at how far away these transcription factors are binding from the genes, and they can still regulate gene expression. And once we started doing ChIP-seq of the same transcription factor in different cells, what we noticed is that very often the same transcription factor, even though they are supposed to recognize the same DNA sequence, in different cells, you look at estrogen receptor binding in liver, in breast, in ovary, in brain, you can see actually the overlap can be pretty small. And you're wondering if it's only sequence determined, how come there is a difference? And so epigenetics naturally become a question that we got interested in. And this is the area that's really under a lot of investigation. So epigenetics, there are different levels of definition. The top definition is heritable changes in gene function that occur without a change in the DNA sequence. So as we mentioned, how come not all the motif sites are bound by the same transcription factor, right? If it's all recognizing the same sequence, why does the transcription factor only go to a minority of sites in the genome that are bound by the factor? How come the same transcription factor bind to different locations in different cells? How come the transcription factor binding only regulates some of the nearby genes, but not other nearby genes? And so this goes to the different level of epigenetics. So the very first, people think of DNA, in fact, in bacteria, this is pretty much all you need to know is the DNA sequence. But once we are talking about eukaryotes, the chromatin, we talk about chromatin, which is DNA packaged into three-dimensional structures. And so the very basic structure of the chromatin is this structure called the nucleosome. It's basically a bunch of proteins in the middle. These are histone proteins, and the DNA, you can see that just wrap around it. And it's usually 150 base pair long, roughly, about 2 circles. And then it will move to the next one. And so if you zoom out a little bit, you can see this is like a pearl necklace, right? And within here, there are 8 histone proteins stacked in 2 layers, and there are 2 rounds of DNA circle around them. And so you can almost think about it as an Oreo cookie, or a pearl on the necklace, right? And so this is this, the nucleosome, or when you see multiple nucleosomes on a string of DNA, it's like a bead on a string. And these things are also commonly stacked together. And so when they are more compact, you can see this. And then you keep on zooming out, you will see something like this. This is what people call 30 nanometer fiber, a more compact chromatin. And then you keep zooming out, you will see these 30 nanometer fibers stack on top of each other, and then they will form something like this, which is the active chromosome. And then when this is even further stacked during the metaphase of cell division, you can see really highly stacked structure, and that's really part of a chromosome. And so what we think about transcription factor binding is really, initially it's just finding the chromosome and then kind of go in, really zoom in, eventually binding to the DNA. And all of the structures above influence how compact the chromatin is and whether the transcription factor is able to find this location and bind. And in fact, in a ChIP-seq experiment, remember the very first step is we cross-link the DNA to the protein, and then we do sonication. And that sonication step was never even. Because if you just have DNA you sonicate, you sequence, versus this structure, with chromatin you sonicate, you sequence, you get a very different distribution, suggesting that, as we mentioned before, if you have a highly twisted rubber band and when you are trying to chop it up, you chop in different distributions as compared to a very straight piece of rubber band, right? So that's the difference. Yeah, so this is kind of more at the functional relevance, functionally relevant changes to the genome that do not involve a change in nucleotide sequence. And there are also people who study heritable changes in gene activity that are not caused by changes in DNA sequence. These are transgenerational changes. For example, during World War II, there's a major siege of German troops around Netherlands city, and there's the resistance were in the city, and they, for a very long time, starvation. And later on, it was discovered the kids of those parents who survived are more likely to have diabetes. And so this is not a genetic change because the parent is just experiencing something, right? So, but it's not really the changes on the parent DNA. It's not the changes on the DNA sequence on the sperm or the egg, but somehow there is a transgenerational difference. And so people are now also thinking about the study of stable long-term alterations in the transcriptional potential of a cell that are not necessarily heritable. So a few interesting phenomena of epigenetics. For example, in a beehive, the queen bee will give birth to many baby bees, and they are put, the worker bees will put these in the larvae, and they will try to feed the larvae with different food. So if it's regular food, the baby bee will become a next generation worker. But if the larva is fed with the royal jelly, they will become a queen. And so this is just upbringing making a difference. And there are some cases when in ant colonies, a similar situation. And interestingly, sometimes if you take out the ant queen, there will be several other queens that will start to fight. And then when their whole body physiology will change, and then they secrete hormone to suppress the other female from becoming a queen, and they will continue to be workers. So you can see all of this is quite interesting. They change behavior, it's heritable, but it's not really on the DNA sequence. There's another example for this agouti mice. So basically the mother mice are the same, genetically identical, but during the pregnancy, depending on what type of diet you give to the mother pregnant mice, the babies would be quite different. For example, so both of their mothers were fed with the same thing, except just one difference in the mouse, in the diet. So this mother received a supplemented diet with folic acid and so vitamin B12, and you can see this is a pretty normal mouse. And so in the phenotype it will have a brown fur color, and it's pretty healthy and it's lean. Whereas the agouti mice is without the folic acid supplement and also low in methionine. So by the way, in the thin mouse, in this region of the DNA is methylated. Whereas in the other case, without enough methionine in the diet, the baby after birth, you will see they have a yellow fur color, and also they are prone to cancer and diabetes. They're also very, very fat, and so very different phenotype. So the mother, by the time of pregnancy, the genetic makeup for the baby, the fetus, is already determined, right? And it's only the nurture part, not the nature part, that's making a difference in these phenotypes. And you could see that the diabetic mice can also potentially give birth to future generations of diabetic baby mice. And so epigenetics is playing a role somehow. You almost feel like Lamarck may not be completely wrong. There are certain things maybe experience will be inherited. It's not completely random and selection. So yeah, this is in human, also nurture versus nature. We all know that zygotic twins have the the same DNA, right? They have identical DNA genome, but very often they have different personalities. And because sometimes when you have a twin, they kind of compete for the mother's nutrient in the womb. And so some would be bigger than the other, and their whole personality could be different. Another example is this: in 1998, in Canada, there is a big ice storm. And so for a week in northern part of Eastern Canada, there is a massive power outage. And so it's discovered a few years later that the kids whose mother were pregnant during this big ice storm, their kids are actually getting a lot of trouble at school. They're kind of more disobedient, and so that's again some upbringing. During the pregnancy of the mom, the stress in the environment created something on the kids that is potentially also inheritable, but it's definitely not on the DNA. And so this is really getting a lot of attention. And so with epigenetics, so this is more on the transgenerational difference, but on the molecular level, this is a epigenetic landscape that is proposed by Conrad Waddington. And so he proposed that in the very top, when you have an embryonic stem cell, it's pluripotent, it's like at the top of the mountain. And the cell has the potential to differentiate into every possible subtypes, right? But then as the cell differentiate, at some branch point, it will have to make some decisions. Does it want to go to the hematopoiesis side, or does it want to develop into muscles or some bones or different organs or tissues, right? So the cell has to make some decisions. And once the decision is made, very often, the cell will just keep going down. For example, if a cell at some identity already decided that it's going to make blood, any kind of like a red blood cell type or white blood cell type, it knows that that's what its identity is going to be. It's difficult for that cell to become suddenly a brain cell, right? So this is really a differentiation kind of in a more at some point you make a decision, but after that it's more deterministic. So the next child will at least know that it's part of the blood lineage and not a epithelial cell. And so depending on the path they go, the cell will eventually be differentiating in different cell types in different organs. And what is really fascinating is now there are approaches such as induced pluripotency. So you treat the cell with some drugs, and some of these differentiated cells can now go back to the top of the mountain again and become another embryonic stem cell. And there are also cases where the figure is drawn like this, it's going somewhere, and then the cell will become cancerous. The way I think about this is like you go skiing and you go from the top of the mountain, you follow different trails, and cancer is you get stuck in a ditch or something, right? So it's going to a place that it's not supposed to go, but it is stuck in some kind of a epigenetic landscape, a ditch that it can't get out. And so epigenetic, you can think about this is what the genes are expressed in a particular cell is like the kinetic energy of a cell. It really determines the state. But in order to maintain that state, you also have the potential energy of the cell, which is the epigenetics. And the daughter cell always know its identity because of the epigenetic status of the cell. And so it's very, very interesting, in development, in disease, in this IPS process. Understanding the epigenetic landscape is very, very important in understanding gene regulation and a physiological process. Questions about epigenetics in general?